Good morning, everyone. I want to thank the organizers for giving me the opportunity to participate in this meeting. And I am going to use PowerPoint presentation because I could never pull off a talk like Dr. Searle just did without this crutch, so forgive me. I spend most of my work with most of the research using monkeys and rats as a way to gain insight into the organization and storage of long-term memory. Now, one of the structures that features prominently in most of my research is a structure known as the hippocampus. The hippocampus shown here, dissected out of a human brain and inverted to give you an idea where the hippocampus gets its name. It's Latin for seahorse. Most of what I'm going to be talking about today is, well, what I hope to convey to you today is that the hippocampus and associated cortical regions provide a particularly robust example of the connectedness of cross species, particularly with respect to the homology of brain function and the organization of long-term memory. So what I'm planning on doing today is to talk about the successful development of a model of human memory and human memory impairment in the monkey and the rat. Now, the promise of using animals to do this sort of thing, to gain insight into human memory, really only makes sense when considered in the light of evolution and evolutionary theory. So the motivation here is that by studying animals and learning about them and learning about their brains, we can gain valuable insights into our own brains and the human condition. But I thought it might be interesting to start today by describing a historic scientific controversy that challenged this very claim, and challenged it on the basis of asserted neuroanatomical differences between the brains of human and the brains of other animals, particularly non-human primates. So this is a picture of Thomas Huxley. Huxley was a noted biologist, paleontologist, and anatomist. Among his many accolades, he served as the president of the Royal Society, the president of the Geological Society, also the president of the British Association for Science, so a very established scientist. However, today he's perhaps best remembered for a heated exchange that took place at a debate that took place just after Darwin's book on the Origins of Species was published in 1859. So this was a colloquium that was organized at the Oxford University Natural History Museum 7 months after the publication of Darwin's book. And a number of scientists and philosophers participated in this meeting, but what was best remembered was a heated exchange that took place between Huxley and Bishop Samuel Wilberforce. Now, in this exchange, Wilberforce asked Huxley whether it was through his grandfather or his grandmother that he claimed descent from a monkey, to which Huxley replied that he would not be ashamed to have a monkey as an ancestor, but he would be ashamed to be associated with a man who used his great intellectual gifts to obscure the truth. So that was the take-home message from that particular debate. But what I want to relay today was not between those two men, but between this man and the man who, in fact, had coached Bishop Wilberforce for that Oxford debate. This Bishop Wilberforce there. Richard Owen. So Richard Owen was a paleontologist and comparative anatomist, and he is best known perhaps today by coining the term dinosaur, but also for his vigorous, outspoken opposition to Charles Darwin's theory of evolution by natural selection. So in 1859, probably spurred on by the impending publication of Darwin's book, he published a reclassification of mammals. Here's the book cover, Classification and Geographical Distribution of Mammalia. Now, in this, he reclassified humans into not only a different order from other non-human primates, but into a entirely different subclass. The new subclass he entitled Archencephala, which means ruling brain. So of course, up to this point, humans had been grouped with other primates into the order known as primates. He was attempting to change all this. And just in case the reader didn't appreciate the significance of this reclassification, on the very next page, he had this statement: "Thus man fulfills his destiny as the supreme master of the earth and of the lower creation." So he was really trying to push a point here. And he based this reclassification on 3 neuroanatomical criteria. The most important of which, the central criteria, was that only in humans does only in humans does there exist a hippocampus minor. And so actually, because most of the talk I'm going to be focused on the hippocampus, it intrigued me that in fact the hippocampus minor was used as a reason to not accept the theory of evolution. So essentially what the hippocampus minor is, this is an 1863 slide that was provided by Huxley, and it's a protrusion in the floor of the lateral ventricle. I pointed to it, it's hard to see, so this is provided by Huxley. And essentially, what the hippocampus minor is a protrusion that's caused by the calcarine sulcus, and it causes a bump on the floor of the lateral ventricle. Now, Owen claimed that only humans had this, but Huxley was quick to point out in this illustration that also chimpanzees have this, and that of course, this would tend to dispute what Owen had suggested. But here's a more modern view of the hippocampus minor. So this is a human brain with the temporal lobe and the parietal lobe dissected away, so you can see the lateral ventricle here. Now, within the lateral ventricle, there are 2 identifiable structures. One is the hippocampus major, as it was called back then. Now it's simply referred to as the hippocampus. And here is the hippocampus minor. Before the 1860s, it was called the calcar avus because of its resemblance to a cockspur. And that's the current use today. So this is the basis for Owen's suggestion that humans were somehow separated from other primates. Well, Huxley immediately realized that this was probably an error, and he began dissecting every sort of primate brain he could get his hands on. And it became clear to him very quickly that these criteria that Owen had come up with were not going to hold. And so in that sense, this debate, this controversy, if you will, was insignificant or anticlimactic. In fact, when Huxley in his later writings refers to it, he often says with the hippocampus minor controversy, if you could call something a controversy where all the facts were on one side and nothing but empty assertions on the other side, a controversy. So he was very dismissive of it. But in another sense, it was an important debate because some authors have argued that not only did Huxley go to great effort to show that Owen's was wrong, but he attempted to show that he must have been intentionally obfuscating the truth. And he does it by a variety of ways, which are pretty compelling. And in doing so, Huxley seriously damaged the reputation of Owen, who was perhaps the most prominent anti-evolutionist scientist of the time. So in that regard, this interaction paved the way for greater and more rapid general acceptance of evolution by the scientific community, which is generally thought to have been achieved, including for the evolution of man, by about 1890. Oh, yeah, so let me go back here. So, and going back into these old records, it's really amazing the amount of satirical comment you can find on this particular debate. Cartoons, poems, and so forth. And I can't resist reading my favorite poem concerning the hippocampus minor controversy. But before I read it, there's 2 other pieces of information I need to give you. One was that in addition to the hippocampus minor, Owen said that the human also only humans had a posterior horn of the lateral ventricle, so extending backwards. And also only the human brain had an occipital lobe that extended so far posterior that it completely obscured the cerebellum. Now, both of these claims were also easily proven to be false, but I mention them here because the poem accurately alludes to them. Now, this poem was initially published anonymously, but a few years later it was shown to be the work of the eminent paleontologist Sir Philip Egerton. It says, Owens, you can see the brain of chimpanzee is always exceedingly small, with the hindermost horn of extremity shorn and no hippocampus at all. Next, Huxley replies that Owen he lies and garbles his Latin quotation. The facts are not new, his mistakes not a few, detrimental to his reputation. To twice slay the slain by dint of the brain, Huxley concludes his review, is but labor in vain and productive of gain, and so I'll bid you adieu. This is really interesting because everything he's alluded to here is exactly accurate in this controversy. And this is actually only an extract of the poem, or an excerpt. The beginning parts talks about Robert Chambers' book, Vestiges of the Natural History of Creation, which was a very inspirational book to Darwin. And it also spoke glowingly of Darwin and his theory of natural selection, so it's really quite a poem. I've only sort of touched on this controversy in the interest of time, but if you read it closely, you see there's very interesting personalities involved and quite vicious personal attacks between the two men that were uncommon even for the standards of the day. And if you're interested in a sort of deeper, looking at this a little bit deeper, I could recommend two really eloquent articles by the neuroscientist Charlie Gross and fellow Academy member. And it's also worth reading Owen and this reclassification. And anything by Thomas Huxley is worth reading. But this 1863 book is where he sort of summarized men's place in history. But he did it by collecting a series of lectures that he was giving to the laymen. So this is really meant for the layman, and it's a very interesting read. So I would highly recommend it. So in moving now from the minor structure on the floor of the lateral ventricle to the major structure, the hippocampus, this story begins with the noted amnesic HM. Now, HM will probably be very familiar to many of you. HM had intractable seizures that grew worse with time, so much so that he was no longer able to live a normal life because of the severity and frequency of his seizures. And so determination was made that he would undergo a experimental surgery in an effort to relieve those seizures. He was on anti-seizure medications, but the doses were becoming so high they were becoming toxic. So they felt like they had no other choice but to try this experimental procedure. So the person who did the operation was William Scoville, shown here. And essentially, this is what was removed. This is a normal brain. You can see the temporal lobes here. And they went into a supraorbital approach, lifted the frontal lobes, and excised the medial aspects of the temporal lobe. So this surgery was, in a sense, successful because it did reduce the frequency and the severity of the seizures, but it left HM profoundly amnesic. So his memory loss could be characterized as profound forgetfulness. And he was studied then, from that point forward, by Brenda Milner, and basically this 1957 paper is considered by many to be really the beginning of the modern era of the neuroscience of memory. So the characteristics of the memory impairment that HM exhibited are the same characteristics that have been observed time and time again in other patients with similar damage. And that includes impaired anterograde memory. So these patients have an inability to form new, lasting, long-term memories. They have impaired retrograde memory, which means they've lost information that was normally acquired before the damage. And it typically takes the shape that was referred to as temporally graded retrograde amnesia, so that the information that was learned just before the damage is gone, whereas information learned long ago is relatively preserved. And this makes the point that the medial temporal lobes are not the ultimate repository for long-term memories, and it also makes it clear that once something is learned over time, there's a tremendous amount of reorganization that must take place, so that initially memories are critically dependent on these structures in the medial temporal lobe, but over time, something happens to make them independent. Now, these really dramatic impairments occur against a background of intact immediate or working memory, intact cognitive abilities, motivation, personality, and intelligence, and perceptual abilities. Now, up to this point, no one knew whether that was actually going to be so or not, and I think a lot of people might have suspected that intelligence and personality would have all been neurologically wrapped up in the same space so that you couldn't separate one from the other. But this makes the important point that memory is a distinct cerebral phenomena and that it's critically important on these structures in the medial temporal lobe. So I just wanted to give you guys a flavor of what this impairment looks like. This is a little film clip from American Scientific Frontiers. It was hosted by Alan Alda, and this is of patient EP. EP had his medial temporal lobes carved out bilaterally by viral encephalitis, which left him profoundly amnesic. And this first clip is a little too closely edited, I think, so you can't just watch it and get a sense of it. So I'm going to tell you what you're going to see, and then I'll run it. The next clip is more informative. But essentially, Jen, the technician here, is reading him a short list of words and just asking him to repeat the words to her. No audio.
What you gotta add is about 2 more.
Oh, really?
Yeah,
Would that make it tougher?
Then that would... Dog, raccoon, lion, and a parrot.
Good.
But he even has a little sly fun with Jen along the way.
What you gotta add is about 2 more.
Oh, really?
Yeah,
Would that make it tougher?
Then that would catch him. 7, you know.
Right, you see this neural damage doesn't seem to have affected his personality or intelligence, and clearly his immediate or working memory is left intact because he can remember the words that she read to him and repeat them back. I think this is 15 minutes later.
I read you some words about 15 minutes ago. 4 of them. 4 words. Do you remember me reading you 4 words?
No, I can't
No?
recall. I'm sorry.
No, that's okay. Don't need to
My
apologize.
mind is getting old.
That's okay. I read you 4 animals, and I want you to tell me yes if you think the animal was on the list I read you about 15 minutes ago, or no if it was not on the list.
Wonderful.
Okay? Cat.
Yes.
How sure are you?
I feel reasonably sure on that
Reasonably
cat.
sure? Uh-huh. Raccoon.
No. No, I feel no.
Giraffe.
Yes.
How sure are you that I read
I'm
you
real
that?
sure on that.
Real sure?
They're beautiful animals,
Okay.
believe me.
Lion.
No.
Okay.
Okay, so you can see this is not a trivial memory impairment. So as soon as HM was first described, scientists knew immediately that they would need to develop an animal model in order to determine what the critical structures are in the brain that were responsible for this sort of a memory impairment. Because remember, with HM's lesion, it included many structures, the cortical areas of the medial temporal lobe, the amygdala, and the hippocampus, and so forth. So at this point, nobody had any idea what were the really critical structures, although there were some historic reasons to suspect it was the hippocampus. No one could tell that for sure. So the obvious solution would be to develop an animal model to be able to test these ideas. And in fact, the person who did the surgery on HM, Scoville, immediately within a year had gone up to Montreal and done the similar sort of surgeries in monkeys in an attempt to begin to establish this animal model. But as it turns out, establishing an effective animal model of this phenomenon turned out to be a torturous task. I want to read you a quote from a review of the literature that was done a full 10 years into the effort to try to establish an animal model. This was an article that was published in Psychology Bulletin in 1967, 10 years later, where Douglas reviewed all of the rat and monkey literature that had attempted to relate findings to this human condition. And it's a little bit of a long quote, but it really does give you a sense of where the field was 10 years into this effort. Hippocampal lesions obviously do not impair learning in general, even when learning involves retention for long periods of time. Thus, the animal and human data would appear to be in contradiction. This contradiction could be resolved by postulating that the hippocampus has a different basic function in man and beast. Such a solution to this problem is generally unacceptable to physiological psychologists, however. Another possible resolution of this paradox is that recent memory loss in man is a secondary effect of a different type of primary disorder. The author has chosen the latter course and suggests that recent memory loss in man is a genuine phenomenon, but that it is a byproduct of interference during storage and not due to a lack of storage ability per se. So after 10 years, the animal folks were not even convinced that it was a true memory problem of the kind I just showed you with EP. They were postulating it must be some sort of catastrophic interference effect. Now, the reason it took so long to develop an animal model is because at the time it was not appreciated there's actually multiple forms of memory, and that the type of memory that the medial temporal lobe damaged was only one type, referred to as declarative memory. So declarative memory is what we think of in the ordinary use of the word memory. It can be brought to mind and declared that, yes, I have this memory. So in other words, you're consciously aware of this type of memory content. Another hallmark of declarative memory is that it can be acquired very rapidly and expressed flexibly. So if I read something one time in a book, I can then repeat it verbally later. That's a flexible expression of that learning, or change my behavior based on what I've read. So it's flexible and acquired quickly. It's generally divided into two types, semantic and episodic. Episodic is just simply personal memory. You remember what you had for dinner last night and all the details associated with that dinner. And semantic is more just fact knowledge. You remember what the term dinner means, but you probably have no clue where you first learned that. And it's this type of memory that depends on the structures in the medial temporal lobe. Now, this can be contrasted with a whole collection of what's referred to as non-declarative memory. Now, non-declarative memory is not available to conscious recollection. So you're not even aware of having this memory content, and the only reason that we know you have it, or that you know you have it, is because you see a change in behavior. So it's expressed as a change in performance because of experience. So because it's experience dependent, it qualifies as memory, but not the type of memory that we can bring to mind and remember. And so there are different forms of non-declarative memory that depend on a variety of different brain structures, which I've listed here. But the important point for this talk is that what wasn't appreciated then, but is appreciated now, is that memory tests that humans approach as a test of declarative memory, animals often will not approach the same test in the same way. So if we were given two objects that we were asked to remember, we would just simply memorize the pointer. Okay, that's the one I'll be rewarded for. Most animals, including monkeys, don't learn things that way. They learn it more over a course of many trials where they develop a bias towards the positive stimulus. And this is often referred to as habit memory, and we know it depends on the striatum. And so the real difficulty is that what the test they were giving the animals seemed like they ought to be assessing the same type of memory that was tested in humans, but in fact they were not. So the real trick, or the real breakthrough, was developing tests for animals that really do measure this type of memory. And that occurred in 1978 with Mort Mishkin's publication, where he prepared animals with lesions just like HM, so these are large lesions of the medial temporal lobe, and he tested them on a test which hadn't been used previously. Well, one other study had used it, but essentially this was the key. It was the delay non-matching sample task. So in this task, an animal is trained to displace an object where it can receive a food reward. Then the object is taken away and a delay interval is imposed, and then the animal is given a choice between the object that's seen before and a new one, and he's rewarded for choosing the new object. So in order to train an animal to do this, you first have to train them over many, many trials to learn this non-matching rule. Once the animal learns to remember the objects that it's seen and goes to the next object, then you can start giving him unique trials each time. So every new trial, there's different objects. So when you do tests like this, the animal never has the opportunity to develop a bias over many trials towards one object. It has to learn what object it is with a single trial, and that turned out to be the key. So there's no graph from Mort Michigan's 78 paper, but here's a study from Stuart Zola's lab where monkeys with large medial temporal lobe lesions were tested on the delay non-matching to sample task. And there's really a lot of important information here. First, when the delay was short, the animals with medial temporal lobe lesions were absolutely normal. And this tells you a lot. It says, number one, the animals have intact short-term or immediate memory because they can bridge an 8-second gap. It also shows that they have the perceptual ability to make this discrimination between whatever the two objects were. It's only when you increase the demand on long-term memory by increasing the delay between when the animal first sees the object and when he's given the choice, and then you see this rapid decline. Now, once you have that with the medial temporal lobe lesion designed to mimic HM, now you're in a position to start doing more discrete lesions to see what are the critical structures and what are the structures that aren't as critical. And this just shows what happens when you do a selective hippocampal lesion. Now, up to this time, everyone thought that the most important player for all of this, and maybe the only player, was the hippocampus. But when you do a selective hippocampal lesion, you do get a delay-dependent impairment, but it's not as substantial as the full lesion. So this immediately showed that, well, there's some other structures that are play important roles, and so then the hunt was on to identify what those structures were. This is a complicated graph, and I don't want to spend too much time on it. It's really meant just to sort of give you a flavor of how you can systematically go through these different brain structures using this animal model. And so this is normal performance on the delay on matching to sample task, and one thing that was learned is if you do a selective amygdala lesion, which was part of that initial lesion in HM, you don't really have any effect on this task. It doesn't affect declarative memory in that way. However, if you do a hippocampal lesion and add the cortical damage around the hippocampus, then you get a more substantial impairment. If you add an amygdala lesion to that, you don't change the behavior at all, further ruling out the hippocampus as important. And then if you lesion the hippocampus and extend the cortical damage forward, you get a full-blown effect. So in this way, scientists were beginning to appreciate that not only is the hippocampus important for this type of memory, but that the cortical areas surrounding it are also important. And of course, in the early 1990s, the rat folks got involved, and so this was a task developed by Dave Mumby, and it's an analog of the monkey delay non-matching to sample task. So it's the same thing. The animal is trained to non-match the sample that displaces an object, then it's retained in the middle, all without handling, so it's not distracting, and then the animal is given a choice to non-match, just like in the monkeys. When you do that with selective hippocampal lesions, again, you see intact performance when working or immediate memory can be used, and impaired performance at the longer delays when the animal must rely on long-term memory. So the delay non-match, the importance of the delay non-matching sample task can't be understated, but it's a difficult task to train. You have to train monkeys a lot to get them to initially learn the task, and for rats, I can assure you it's even more difficult. As it turns out, also in the early 1990s, a different sort of task, it was discovered that a different type of task could also assess the same sort of memory as the delay non-matching to sample task, except it was much easier to administer to an animal. And this was called the visual paired comparison task. So essentially, you just show two objects that are exactly identical, then insert a delay, and then show a picture like this. And what many of you may have noticed doing is when I put this slide up, you probably initially spent a little more time looking at this than this, probably about twice as much time. And the idea here is, well, you remember this, it's familiar, and now it's less interesting, so you're attracted to this. And you can measure this in humans and monkeys in exactly the same way. You just simply show these objects, and you just measure where they look. And it's quite simple to do. It doesn't require any rule learning and verbal instructions. It could be administered to monkeys and humans in exactly the same way. When you do that, you see that humans with damage that includes the hippocampus again show this delay-dependent impairment on this task, and monkeys with selective hippocampal lesions also show the same sort of memory impairment profile. You don't have to change the task much to administer it to rats and monkeys. Only in the rodent, the animals are allowed to physically explore the object for a period of time, and then you just simply swap out one new object, and if the animal remembers this one, it'll spend a disproportionate amount of time explaining this one. And when you do that in rats and monkeys, again, you see the same sort of behavioral impairment profile with animals that have damage to the hippocampus, damage or disrupted to the hippocampus. So this, in effect, gives us confidence that what we're measuring in animals is the same thing that we're measuring in humans. This is a task that is administered essentially the same way to the different species, and the same impairment profile results with the same type of brain damage. This is just a quick example of how this task can be used to identify other structures in the medial temporal lobe that are important. So this is control performance. You saw the memory lasts a good amount of time, even with a single exposure. When you're losing the perirhinal cortex, you see intact performance at the shortest delay, but again, increase the demand on memory, you get this impairment, suggesting that now it's not just the cortical areas around the hippocampus, but now we're beginning to see exactly what cortical areas they are, perirhinal cortex in this case. When you move the lesion more laterally to visual area TE, which is a unimodal visual area, which projects heavily to the perirhinal cortex, you get something that looks like this, which the animals never perform above chance, presumably because they now no longer have the perceptual hardware to make a distinction between the two objects, so they're never going to be above chance. Of course, it could be argued that this is just a really profound memory impairment, but these monkeys were then later tested on to delay non-matching sample, but not with visual objects. They were trained to feel an object as a sample phase, and then during the choice phase, felt two objects, one that was the same and one that wasn't. So this is a memory test, but it doesn't involve vision, and these animals performed absolutely normally on that test. So again, this just shows how now the TE is excluded from this memory system. So I think by any definition, this model of human memory impairment in the human with non-human primates is a success story, and these are just some of the characteristics that have been successfully modeled. Memory is impaired on several tasks, including those that are identical to the ones given to humans. The impairment is exacerbated by increasing the retention delay or the amount of material to be learned. It's exacerbated by distraction. It's not limited to one sensory modality. Impairment can be enduring. There's a retrograde effect. I haven't talked about any of that, but that phenomena has also been demonstrated in the animal model. And this occurs against the background of spared skill-based memory and immediate memory. So this is a success story. We now know that the critical structures are the perirhinal cortex, the perihippocampal cortex, the entorhinal cortex. I show them highlighted here. These, of course, are also anatomically connected to the hippocampus, which you can't see. These same structures are easily identifiable in this ventral view of the monkey brain, and they're also there in the rat. It's named different in the rat. This is more anatomically descriptive. It's called the postrhinal cortex in the rat because it's posterior to the rhinal sulcus, but essentially the organization is the same between all three species. We know now that this memory system is a hierarchically organized system where information from the neocortex, particularly from visual areas, goes to the perirhinal cortex. More spatial areas goes to the perihippocampal cortex. This information remains relatively segregated as it enters the entorhinal cortex. Spatial information going medial, more visual information going lateral, and then the entorhinal cortex sends information through the dentate CA3, CA1, and subiculum in this sort of feed-forward loop that's often referred to as a trisynaptic loop. And the important point here is these colored structures. These are the structures that, when damaged, causes an impairment in declarative memory. And I should say that now this is very well accepted, and there's a great deal of work going on to try to determine what portions of the memory these different individual structures contribute, but that's ongoing. But there are challenges to this work, and I really alluded to one of the big challenges which prevented the quick establishment of an animal model, and that is humans often will approach solving tasks differently from other animals. And so it becomes a question of how to relate the findings from animals to humans. And one question is, humans are predisposed to use declarative memory. There's no doubt about that. This begs the question, do they even have a fully developed non-declarative memory system that can acquire tasks like animals can? Well, that question was addressed by Larry Squire and his colleagues recently by using this concurrent task. So this is a task that humans solve by memorizing, using declarative memory. There are 8 pairs. One of the pairs is always the plus, and they're given sequentially, and usually about 5 times. Each pair is presented about 5 times a day in random order. Now, humans memorize this, and they also learn it fairly quickly. This just shows control performance across sessions, whereas amnesic patients don't learn this task very well. They struggle because they have an impairment in declarative memory, and that's the way humans approach this task. Whereas if you look at exactly the same task in monkeys, you see that they take hundreds of trials to learn it. This is normal performance, but that animals with hippocampal lesions learn it at the same rate. So this was really what confounded the early attempts, is because this looks like the lesion had no effect on memory. In this particular study, some of the lesions went from the hippocampus and encroached upon the caudate nucleus. And we know that, we think that the caudate nucleus is important for a habit type of memory. And when that happens, you do see an impairment on this task. So a question becomes, if humans without declarative memory were tested in the same way the monkeys were, would they even be able to learn this task at all? And this was recently conducted. There's a EP who you've already met. This just shows his medial temporal lobe lesions, and another patient, GP, also with viral encephalitis. They were essentially given this 8-pair concurrent task, but instead of 3 sessions, they were just tested repeatedly until the experimenters either gave up or saw some evidence of learning. So in this particular study, this is how control performance is very fast. They learn it rapidly. And one of the hallmarks of declarative memory I mentioned earlier is that it can be expressed flexibly. So after the humans have learned this task, instead of giving them individual discriminations with the objects, all the objects were piled on the table, and the subjects were asked to sort them into the correct ones and incorrect ones, which they did without any problems. And then, of course, when the test was reconstituted, their performance was just fine. Now take EP's performance on this test. So he did not learn this task in 3 sessions, but over the course of 36 sessions, you see this gradual increase in performance. So EP began learning this task, and this is at a rate that's not dissimilar from what we see in normal monkeys learning the task. So he was able to acquire the task. Now the question becomes, can this knowledge, which is clearly acquired here, be expressed flexibly? And so when EP was given the sorting task, his performance fell to chance. Okay, so utterly unable to do that. In fact, he didn't even sort the correct and incorrects into equal number piles. When the test was reconstituted, however, he performed just fine. And when he was brought back 17 days later, the results were still the same. The results for GP were exactly the same. So this shows that humans who essentially have no residual declarative memory can nonetheless learn the task non-declaratively. And in fact, I think it's instructive for me to relate to you some of the comments and some of the behaviors that these subjects were engaging in. First, the objects all had the word correct or incorrect on the bottom of them. After about the 4th session, both patients began to spontaneously pick up the objects and look underneath, even though they couldn't give a good description of why they began doing that. At one point, EP said to the tester, how am I doing this? And when the tester asked EP, well, how do you think you're doing it? He says, I don't think it's from memory. I think it just seems like it's going from my head into my hand. GP also said that the correct object just seemed to pop out to him. So in this sense, this provides a real nice dichotomy between declarative memory and non-declarative. Subjects can learn this task very well, but they absolutely do it in the absence of any awareness of having learned the task. In fact, before each testing session, the patients were asked to describe, do you recognize these objects? They always said no. What do you think we're going to do with these objects? They generally said something like, well, you're going to show them to me and we're going to have a discussion about them. So they really had no sense of what they're even being used for, despite the fact that they were performing as well as controls by the end of these many sessions. So I think, in summary, it shows how there are clearly difficulties or differences between species, but in many cases, these differences can be overcome. The animal model represents a success story, but when interpreting results from animals to humans, there are complications. But I think these complications can be mitigated by understanding that different species have evolved under different selection pressures, and that may bias them to approach the same task differently. But by understanding that these differences exist, you can use sophisticated behavioral analyses to apply to the problem and in many cases, get a clearer understanding of what first appears to be confusing results. Let me just finish by thanking my longtime collaborator, Larry Squire, who had a very influential hand in all the stuff I just presented, and all of the other collaborators and students that participated in these projects, particularly Stuart Zola for the work with the monkeys. Thank you.